Hello, my name is Emily, and this is my presentation on the COVID-19 testing process. 2019 novel coronavirus, or COVID-19, is the name of the disease caused by the severe acute respiratory syndrome coronavirus 2, known in short as SARS-CoV-2. There is a viral test and an antibody test for COVID-19. The viral test is for current infection and uses a real-time reverse transcriptase polymerase chain reaction. The antibody test may be ordered online or through a healthcare provider and is used to test for past infection by detecting IgG and IgM antibodies. The RT-PCR test is the more prominently used test right now because it diagnoses acute infection when the disease is still contagious. Real-time reverse transcriptase polymerase chain reaction, or RRT-PCR, is used to test for the SARS-CoV-2 virus RNA in upper or lower respiratory tract specimens, including nasal, nasopharyngeal, oropharyngeal swabs, sputum, lower respiratory tract aspirates, bronchoalveolar lavage, and nasal aspirates. It is a qualitative test, so it will have a positive or a negative result. RT-PCR is used to make copies of short sections of DNA from a small sample so that it can be tested and detected. This process is called amplification. SARS-CoV-2 is an RNA virus, so it needs to be made into DNA before it can be amplified. During the test, reverse transcriptase binds to the virus's RNA and makes a DNA-based copy so that it can be amplified. The process of amplification is achieved through repeatedly heating and cooling the sample in cycles. When this happens, the DNA is denatured and separates into single strands. Then Taq polymerase is used to synthesize two new double-stranded copies of the DNA using the original as a template. If a sufficient amount of viral RNA is present in a sample, it will be amplified and detected, and the test will be positive. If no viral RNA is present, the test will be negative. In a singleplex format, the test uses 3 primer and probe sets to detect 3 regions in the SARS-CoV-2 nucleocapsid gene, and 1 primer and probe set to detect human RNase P. When multiplexed into a single reaction, the test uses 2 primer and probe sets to detect 2 regions in the SARS-CoV-2 nucleocapsid gene, and 1 primer and probe set to detect human RNase P. I found my information for this presentation from LabCorp, which is under the Food and Drug Administration, LabTest Online, the World Health Organization, the National Human Genome Research Institute, and the Centers for Disease Control and Prevention.